Ariana.
Alrighty.
Ariana, we can
Yes.
hear you.
Okay, perfect. All right, so we are going to start. So hello, everyone, and welcome to Native Voices in STEM. I am so excited to welcome you all to the seminar, introduce the speaker for today. First, I'd like to start by acknowledging the land that the University of Arizona occupies. So we respectfully acknowledge the University of Arizona is on the land and territories of Indigenous peoples. Today, Arizona is home to 22 federally recognized tribes, with Tucson being home to the O'odham and the Yaqui. Committed to diversity and inclusion, the university strives to build sustainable relationships with sovereign Native nations and Indigenous communities through education offerings, partnerships, and community service. So I always look forward to these seminars every month, and it's great to see so many different faces and names here today. The Native Voices in STEM seminar series is co-sponsored by NSF NRT Indigenous Food, Energy, and Water Security and Sovereignty Program, or Indigenous for short, the University of Arizona Sloan Indigenous Graduate Partnership, and the Department of Chemical and Environmental Engineering. The seminar highlights Native scientists, engineers, educators, leaders, and social activists. It offers students an opportunity to network and learn about the research and careers of Native leaders in various fields. So today I have the pleasure of welcoming Dr. Cherie De Vore. Dr. De Vore Diné is from Crownpoint, New Mexico, in Eastern Navajo Nation. Her clans are Red Bottom People, Atop the Mountain Towering House, Salt People, and Tangle People. She's currently a postdoctoral scholar in Earth System Science at Stanford University. She received her Ph.D. in Civil and Environmental Engineering at the University of New Mexico under the mentorship of Professor Jose Cerrato. Dr. De Vore was awarded an NSF Earth Science Postdoctoral Fellowship to conduct research under the guidance of Professor Scott Fendorf. Her current research is related to better understanding biogeochemical mechanisms affecting the mobilization and bioavailability of metals and soils and plants near Indigenous communities. So please help me welcome Dr. Cherie De Vore.
I think that one was mine. I need my coffee. I'll just stand here so folks online can see me, and I'm going to take off my mask. So thank you so much for hosting me, for creating this program, and hosting several wonderful voices in this seminar series. Several partners are involved in bringing me here, down to folks that organize all of my travel plans, taking me to lunches and coffees. So I definitely appreciate the hospitality and the support as I jump into this seminar. So I'm going to talk a little bit about some of the mechanistic insights around the mobility of metals from places like those impacted by mining and those impacted by wildfires. But what I really wanted to focus on, especially since the theme of Indigifuse and this seminar series is to really talk about community engaged research, focusing on some of those restorative insights as an Indigenous investigator. And before I do that, I would like to introduce myself with this. Would advance. Next slide. Uh-oh. Let me restart this. Slideshow. Okay, here we go. I always like to start my talks in whatever scenario, academic or not, by introducing where I'm from and my family. So I come from a really beautiful family, and we are from the community of Crownpoint, which is in Eastern Navajo Nation. Ariana did a really great job introducing my clans in English, but in Diné, those are Tsinyaanalk'id. Zilt akia ani bashishin shiche a shehan damle do tkatne sahne dashamale. The kut aestana do oma nishla. I have a picture here of the hogan that our family has in an area known as Tsinyaanalk'id, which is an area known to be a space where families in the Crownpoint community could come harvest wood. So our family's homestead still occupies that space to this day. I've included photos here of my grandparents, both my maternal side and my paternal side. And I do this as an exercise to remind myself and my audience of the very unique relationship that Indigenous people have with education, and that my pathway is what it is because of the foresight of my grandparents and our old people and all of the leaders in our community. So the story of my grandparents is that on both sides, neither one of them were able to attend post-secondary education. Each of them raised like a pretty sizable family, subsisting off the landscape, herding sheep, relying on all of the products in textiles, Navajo rug weaving to support their families. And so coming from an exclusively Navajo speaking household to my position where I'm able to articulate cultural values, family values for my family, but also speak the language of scientific critical inquiry, right? It's important to acknowledge this history because we in this department as a school, as academics in general, are going to come across Indigenous students that would like a space in a place like the University of Arizona or Stanford University, which is where I'm doing my postdoc. And so having some insight about our very unique perspectives is really gonna take us a long way in these conversations about inclusivity, diversity. So a little bit more about my professional background. I received my degrees from the University of New Mexico. So very much a systems-based approach in the undergraduate level in environmental science. I got introduced to research at a young age in that department, which I'm incredibly grateful for. And then decided to pursue a master's degree and attained my Master of Water Resources with a particular emphasis in hydroscience. In other words, hydrology, with some really interdisciplinary insight from disciplines like water law, policy, management, economics. And then that then propelled me to pursue a PhD in civil and environmental engineering. It's important for me, in addition to acknowledging my family, to acknowledge my mentors. And I would not have attained a PhD had it not been for several of my mentors, especially at the University of New Mexico, but in particular, Dr. Jose Cerrato, who may or may not be on the call. If you are, hi, Jose. And the PhD project that arose out of that work had to do with understanding all of the biological, chemical, and microbiological mechanisms responsible for the release and bioavailability of arsenic and those areas impacted by mining. And so now I'm a postdoctoral research fellow in the Department of Earth System Science, and I'm mentored there by Dr. Scott Fendorf, who's just phenomenal. And all of the research group members that I have there are also phenomenal. And finally, all of this is possible through a grant, the National Science Foundation Earth Sciences Postdoctoral Fellowship. So I'm a fellow through the end of this year. I really wanted to share this really nice infographic that was created by a museum in New Mexico called Explora in collaboration with the Institute of Museum and Library Sciences. I think these sorts of infographics are really powerful for communicating to a broader audience, like identity and identity in our science. But very quickly, I wanted to gauge from folks here, at least in person, and feel free to jump on the chat, is this question. When you hear the words mining and when you hear the words wildfire, what comes to mind, like immediately? Anything? Sure.
Actually, I was going to ask you to minimize the participants.
Oh, yes. Yes, yes. Good. That's so helpful. Yeah. at some people in chat. What did they say? Extraction, disturbance, element mobility, ecosystem messed up. Anybody else here in person? I gave y'all chocolate as a nice trade. Scars, thank you, Mark. Wildfire. Yeah. Mine cart sparking and creating hydrophobic soils. Yeah. Grass on fire. Yeah. And contamination. These are all really relevant words as we start to tell the story about how our communities have been impacted, right? And often what's communicated about Indigenous people, specifically in these scenarios where we're both physically and socially isolated, are narratives of a very real traumatic history with these extractive practices. They're communicated from a narrative of despair, of loss, and sadness. And these are very real scenarios. But where I like to counter this narrative is to speak about, okay, we have all of these issues that we're grappling with, but when we can confront these stories with stories of our own, that slide, please. We can start to leverage the tools from our origin narratives as Diné people to speak of the abundance of knowledge, of creativity, of ingenuity that our families and communities have, celebrate all of the milestones and accomplishments that drive us to do the work that's important to us in the first place. So what I've chosen to share today are what have been for me really family and community oriented practices that have reconnected me to our cultural foodways and our cultural repositories around corn, growing corn, food sovereignty, and then understanding how those practices and stories are passed down to our children and the next generation so that they can have the same tools going forward. In addition to that, part of the uplifting of our landscape and being embedded in our ecologies is the ability to heal. When you contribute to the uplifting of others, you find your own healing in that process. So what I've opted to include here are photos of me doing things that like feed my soul. So for example, working with plants. The top two photos are pictures of me preparing herbs that we made for folks that were impacted by COVID. So immune boosting plants that are culturally relevant and that members of our community sought out from our family just north of Crownpoint. And then I have a picture on the bottom right of what we call the netka in close to Tsinyaanalk'id, which is one of our sacred spaces in Diné culture. And then on the bottom left is a picture of myself inside of a steamed corn pit during harvest of last August, where we're able to take all of the harvests for the Navajo white corn, process them, preserve them, and then really give a leg up to folks in the community that want to learn this process, that want to feed their families, and that want to carry on the traditions. So this all to say that the grounding philosophy and the grounding paradigm and paradigm shift for me as an investigator is grounded in community and family. And that's what I really want to challenge each of us in the room, whatever that is for you, not being afraid to weave your identity into the type of science that you do. And I chose to include in the right middle a photo of my previous research group at the University of New Mexico. Jose is right there in the middle, because that space really cultivated like a family-oriented energy, right? Where we're able to create space for folks that are away from their families and find some sense of belonging. And that was critical for me to even like begin and finish a PhD in the first place. So just other photos of friends and family that helped to ground me in this process. Now, I have another question for you all, and I know several folks on the call will know this concept. By a show of hands, how many of you have heard of the river continuum concept? Dr. Chief, Kim Ogden, let's see in the chat. It's really a fundamental paper in the areas of hydrology and ecology. And essentially, the broader idea is that when you think of river systems, and in fact, whatever is relevant for you and where you come from, systematically, if you affect one part of the system, you holistically affect the other parts. So what I drew from this paper that I'm going to use to try to create my own model is, come on. Okay, there we go. That the river continuum concept lends these ideas about a system as being dynamic, moving, and that, as I mentioned before, all changes are interrelated. If you think about the headwaters, all of these high mountainous first order streams that are responsible for introducing a lot of organic matter and leaf litter into the system, which then drives all of the biological diversity in the mid-reaches, which then, when you spread all of those resources out into what we call the lower reaches, the system is working towards a steady and balanced equilibrium, and it's adaptable. Using these concepts, I decided to think about a river is relatable. I chose to include a picture of one of our sacred mountains on the border of New Mexico and Colorado. And this is like an aerial view. So for those headwaters, right, those first order fundamental streams, what that looks like for me is Newtonian physics, thermodynamics, biogeochemistry, all of these fundamental things you learn in your classes, right? Your graduate work and all of your research, along with the fact that we as individuals don't know everything, and that we really have to leverage our collaborations and our connections with other people to do work that we really care about, right? So that stream you can see meets together, and then they all meet with that lower confluence. But as we work down, another big part of this headwater system for me is indigenous knowledge. And I feel like that's really valued here at the University of Arizona, especially within the Indigenous Resilience Center. Our communities are paramount for informing the types of questions we ask in our scientific endeavors. And then for me, especially, all of our place and space knowing, our indigenous philosophy, our family values, as I mentioned in all of my previous slides, are super important. And then in the mid-reaches, where we have all of the biodiversity, right? All of the many things that feed into your system and that can be changed by the system. I like to articulate this as being, okay, for the top streams, those meet, and then you can really start to think about what type of toolbox you want to have. What goes in your toolbox, what tools are needed the most. And in my training, I've really focused on gaining a better understanding of technical expertise using microscopy, advanced spectroscopy, synchrotron-based spectroscopy, biology, culturing, aqueous chemistry, biology, and plant physiology. On top of all of that, your awareness, your social and political awareness, and the social and political climate that we're currently dealing with has a lot to do with how this system is going to be affected, how we are resilient in the system. For my research in particular, energy and extraction is also a very huge reality, but we can also shift that narrative to talk about resource recovery and placemaking. And then that then lends to what types of restorative practices do we want to build on native lands and for our people. And then you'll notice where the streams come together. We call this a confluence here. Really critical part of river systems is that when you take several layers of different disciplines and ways of knowing and experiencing this planet and like carrying knowledge, there's some mixing, there's some responsibility, right, on our parts. And where I think is critical is building trust relationships, not just with communities, but with each other, right? And then developing our pedagogy so that we're always open to learning, even as teachers ourselves, and that we're open to learning from our students and from our collaborations. And then finally, etiquette, how you navigate spaces, especially with things like the public domain and public lands. Oftentimes, we're not really supposed to be in spaces, even though they're public lands, right? So if you're taking samples from a particular place, and as Native people, we have our own way of offering to living biological organisms and then returning those same samples. So having conversations about that and really working within the system to make it more accessible and equitable is paramount. So there's that. Finally, all this to say that for my visit today, hoping to communicate that this toolbox and all of these fundamental paradigms can lead to really awesome mentorship opportunities, but also engineering, in my opinion, is a really powerful tool for social and environmental justice initiatives. And then as an investigator, my goal really in doing science is being able to articulate and communicate the data and information to people that need it the most within a relevant cultural context, and to be able to self-determine with things like data sovereignty, with things like IRBs, and working within correct protocols to do work that's fair. So we have 20 minutes or so. Now, for this next part of the talk, now that I've given you some insight about the type of investigator that I am and the types of foundations I have, is to zoom into the toolbox a little bit and show you what some really unique tools are for adding to a system like this. So folks can specialize at the graduate level and at the faculty level in more community-based work, more field work, more bench scale, micro scale work. But the advantage of starting in a system-based discipline, zooming into an environmental engineering, really like mechanistic oriented discipline, and then coming back full circle into an earth systems program has really enabled me to think big picture about all of the biogeochemical processes involved in the work that I would like to do around environmental restoration. So understanding, there's a photo of a plant at the cellular level, to field work, laboratory work, which I'm very, very familiar with, and then navigating field spaces appropriately. And that, to make the point that something I alluded to earlier, is that before you even begin your research questions and all of your measurable objectives and hypotheses, right? Understanding that if you're working with tribes, paramount is the ability to self-determine, the tribe's ability to self-determine research, and then having appropriate consent. When I entered this space as a visitor on the Atop and Yaqui lands, there's a certain way that I do my own acknowledgement. You'll notice I didn't do one at the very beginning, because I think Native people navigate spaces very differently than non-Native people. Places also have a memory, much like when we work out and we lift weights, our muscle memory kicks into place. Similarly, there's like energetically places that have memories. And so that plays into all of the planning that includes considering historical uses of spaces, how it's currently being used, and then future visions, 7 generations into the past, 7 generations into the future. I've included photos of myself with community members, colleagues, and then the 2 people that I mentored that are both Diné students at the University of New Mexico in applying all of these principles for developing for ourselves when we're writing experimental protocols around etiquette, protocols around specimen collection, doing offerings, and then returning these samples. And then asking the question like, okay, who does the data belong to, right? To give context into the work that I've done with metals mobilization around mining, I started at the master's level doing work at Laguna Pueblo, which is home to the Jack Pile mine. And at one point in history, it was considered the largest open pit uranium mine in the world. And then members of my PhD group worked in Blue Gap Tachi, which is a chapter on the Navajo Nation in central-ish Arizona, around uranium mobility. And then for my PhD, I did a bulk of my work in Cheyenne River, which is Lakota territory in north central South Dakota. All of this to say that it's a really unique exposure scenario in the West for Indigenous people, considering that of over 160,000 abandoned hard rock mines, 3/4 of those are located near federal and tribal lands. And then over 500,000 folks in these states are located within 10 kilometers of one of these features, including myself, including several of my family members. So the big question, in addition to characterizing the extent of the problem, things like concentration, we really want to understand at the mechanistic level, what does it take to get these metals to move in the environment and to make them bioavailable to humans. In addition to that, more recently in my postdoc work at Stanford, understanding what the contribution of metals are to the environment from wildfires. And so here's a photo of one of my sites in Sonoma County, California, one of 4 different counties we're working with at the moment. And this is so important under the current climate change scenarios, considering that 13 of 20 most dramatic fires in most recent history have occurred within the past 5 years. So something we have to look out for, not just in California, but in the West. So I'm going to give you just a snippet of a paper I have in review, trying to understand the coupled release of arsenic, iron, and manganese from mine waste sediments from that area of north central South Dakota that I previously mentioned, and differentiating between what are the chemical and microbiological triggers. So very quick context, I talked a little bit about this the last time I was here, but the driving point of this slide is that upstream of this community, you have arsenic measured in sediments over 2,000 milligrams per kilogram, and the crustal average for this area of the country is around 9 to 10 milligrams per kilogram. When you go downstream on this map to where the gray shaded area is, the reservation, even though it's that far, you have a lot of potential for burial and resuspension along the way. You still see arsenic in the range of 129 to 235 milligrams per kilogram. It's important to note all of that, especially since you have an economy that's incredibly dependent on the grasslands there and on several plants along that riparian corridor for medicinal and food uses. So I characterize using 2 different techniques the sediment in that watershed. On the left, you have Mossbauer spectra, which is a bulk technique that uses an iron 57 isotope to give us information about whether or not iron is oxidized or reduced. And what we found to be in this case is that there's very similar mineralogy between iron at both the upstream and the downstream on tribal land scenarios. And we have iron mostly in the form of goethite. That compared to on the right, X-ray photoelectron spectroscopy measurements, which give us information about speciation of what I have here, arsenic at the near surface. So the near surface being between the first 5 to 10 nanometers, I believe. And we can see that there is on downstream tribal land, which is the DR, more oxidized arsenic in the form of arsenic 5. And then even more so at the Whitewater Creek, which is closest to the Homestake mine. So characterizing these solids, we thought, okay, well, if microorganisms are involved, who's there? What communities are there? So we did some DNA extractions and Illumina sequencing and found that close to 5% of the microbes were arsenic oxidizing or arsenic reducing bacteria. Knowing that, we decided to give them several electron donors. I've only highlighted glucose here, but we've done several that are thermodynamically favorable. And glucose had the most arsenic release under the various redox conditions that we tested. So what you can see here is we have the mine waste solid. Here, I hope folks online can see me pointing with the mouse. An electron donor, put them in these very simple batch incubations for 7 days. And then after 7 days, we have several scenarios. We could keep it from the very beginning completely anaerobic, or at this point, cycle it to anaerobic conditions. And these were reacted over 168 hours. At the end of the 168 hours of the experiment, we collected samples, filtered them, and then did some characterization of the solution. And we found that under the completely anaerobic scenario, we have similar values of arsenic released into solution from those amended with glucose compared to those just reacted with water, right? So on the y-axis, you see arsenic in micromolar over that time period, 14 days. However, if you look on this graph to the right, you see that for the aerobic part of the experiment, very little arsenic was released in the glucose and the water incubations. But once you take away the oxygen from these microbes, they are forced to respire other metals. So in this case, they utilize arsenic, and that's what you see being released into solution. This gray area here, I decided to include in this experiment because I published another paper trying to understand competitive ion displacement from these same solids, and that's what you see here. So this is a different mechanism entirely than what we're describing here. So in addition to what we see in the aqueous measurements, we analyzed these solids at the end of the experiments using XANES, or X-ray absorption near edge spectroscopy at the National Accelerator Laboratory at Stanford. And the more to the left you go in this figure, the more reduced it is. And you can see for this aerobic to anaerobic condition, you have this little shoulder here that says to us that arsenic reduction occurred. So from arsenic 5 to arsenic 3. In addition to that, under those scenarios where glucose was amended, either under a completely anaerobic condition or cycled from aerobic to anaerobic, we had the greatest microbial diversity measured using the Simpson index. Let's see how much time we have. I can rush through this part. I provided the QR code because I figured this would be the case. This is a paper that I published last year. And if you're interested in reading it in its entirety, please feel free to go look it up. And it was chosen as a supplementary cover for this special issue in Earth and Space Chemistry, which is an American Chemical Society journal. So I did this experiment trying to understand the role of endophytic fungi on the uptake of arsenic. So why is that? Why are fungi important? Well, it just so happens that they co-occur with over 70% of terrestrial plants, especially in the latitudes of interest for this study. Fungi are really important in a symbiotic scenario to help plants deal with both biotic and abiotic stresses, such as metals. And they can help plants access vital nutrients like phosphate and organic carbon. So what you see here is seeds that we sterilized and then added an inoculum of the specific isolate of fungi. And this photo here is a microscope image of what that fungi looks like inside of the plant. And we found that when we grew these plants with these fungi, on the top you have plants grown without that are small. Many of them didn't survive the experiments with exposure to arsenic, and then those below really thrived. So when we expose these plants, like the one on the bottom, to arsenic, we can see that within the first, I would say 12 hours of this experiment, 80% of the arsenic was gone from solution, and it stayed that way in this equilibrium for the rest of the experiment. We considered a lot of things. We considered aqueous chemistry, did a lot of geochemical modeling just to verify that complexes weren't formed just because of being in water, but that the plant had something to do with it, right? So where did the arsenic end up? Those without fungi saw that arsenic accumulated mainly in the roots. And those with fungi, there was some translocation and intracellular uptake of arsenic into the above ground parts of the plant. For the scenario that plants were not inoculated with fungi, here is a electron microprobe map of the association between calcium, phosphorus, and arsenic on the root surface of these plants on the top. You can see that the bright spots here represent like more intensity and concentration of arsenic with phosphorus and calcium. But then the same was the case in terms of arsenic, phosphorus, and magnesium in those plants that were amended with endophytes. On the left is a transmission electron microscope image just to verify that endophytes were colonized within the plants. And then the photo is a cross-sectional view. So the red outlines show, like, if you're slicing bologna and you're looking down into your loaf, that's essentially the view that you're getting here. So the hyphae are the red outlines. To the right, we have a stem image of what arsenic looks like inside of the plant. So this bright spot here is osmium, and it's a counter stain when you're working with biological materials, and it tends to gravitate more towards like carbon materials. So, which is what fungi, the building blocks of fungi are lots of carbon chains. So that's where you find a lot of the arsenic inside of the plant, as opposed to just the surface. And that we verified with doing some translocation, really quick calculations, and saw that those with cataphora, or the fungal isolate we were working with, observed some translocation in those plants. So you can communicate the story a number of ways, but there's a restorative framework that I hope folks would take away is that there's potential with fungi, there's potential with what you already have in the environment, and that fungi can help plants tolerate metals. Just a few minutes left. I might skip over this a little bit to get to my wildfires work, but essentially we're interested in understanding what arsenic removal looks like without the complexity of a plant material. So we did some fungal bioabsorption experiments with the broth, and that's what these plates look like when you're growing fungi under the microscope, and then putting those in a broth medium without a plant and understanding arsenic removal. I'll skip over that. So this is what I'm working on currently. I have like some really awesome people in my research group at Stanford, including Claudia Avila, Alondra Lopez, who's a PhD student, and a lot of this groundbreaking work within the past 2 years is based on work that she did. So full credit to Alondra Lopez, who's just like phenomenal, and then collaborating with our mentor, Scott Fendorf. All of this to say that we're interested in what the role is of wildfires on generating toxins, specifically chromium, and what that means for ecosystem recovery after burning scenarios. Here's a map of the counties of Lake, Sonoma, and Napa, which is north of the Bay Area, about an hour or so drive. This map articulates from a low to high burn severity what these wildlife preserves look like. Yeah, so it's important to note that these are protected areas. So they're really great for doing research because the impacts of human activity are minimized. And then you can see in these 3 preserves, we have a mixture of both low and high severity burns. And then this huge one here is another. But for the purposes of this talk, I'll zoom into number 1, which is called Pepperwood Preserve. And it experienced both moderate to high burning severity. It's approximately 3,200 acres and was burned both in the years 2017 and 2019. What's important to note about this preserve is that there are several things that could contribute to metals mobility here, and that has to do with the parent material. So serpentinite or serpentine, if folks are familiar, really enriched with several metals, including chromium. So you can imagine what the impacts might be for metals mobility if these minerals are burned, if they're uptaken into plants, and if that ash is burned. For folks like firefighters, farmers, and people that are making sure we as a country are fed, those questions around exposures are really important. So let's see, just a couple of minutes. I'll go through this very quickly. But this is based, as I mentioned, on Alondra's work, and she did some initial measurements from several of these sites to show that a lot of the chromium-6 that was detected was at the near surface. So within the first 4 centimeters of a lot of the soil cores that were taken under low to moderate to high burning severities. So my question coming into this project was, what are the implications for ecological recovery if we consider things in the environment that we already have, like microorganisms and fungi, right? Dr. Avila is really taking a lot of the leadership in the first question that we have here, which is, how does varying fire severity and vegetation affect ash toxicity? And then my role in this is to understand how microorganisms serve as the basis for recovery and accessing those vital nutrients and changing the chemical composition of the soils, and then the chemical composition of the plants that are burned in these areas. Shameless plug that we have 2 really awesome programs that accept people. Well, the CSIR is from within Stanford, but SURGE accepts people from all across the country. So we just wrapped up our interviews for this program for this year, but always looking for people from all institutions of backgrounds that have some overlap with earth system science type of work. The toolkit for approaching this work, a bulk of it will be done during the summer with our students, because I feel like there's a lot of work to do, but there's also room to spread out those resources, right? In the model that I presented. And that there's room for including multiple types of knowledge in areas like the grasslands of the Dakotas, right? It's not as beautiful as it is because of coincidence. It's because Native people in Dakotas stewarded that land and ensured that low to moderate prescribed burns really balanced the ecosystem there. And similarly, in California, Indigenous people there have always stewarded the land, have always had prescribed burns, and those have a number of implications for ecosystem health. A lot of the burning, the low severity burning really helped to clear sort of the understory to make way for foraging, helped with pest management. Pest management then helps with hunting and the availability of foods to be foraged. So then what we're already observing is that regrowth of vegetation being accelerated. So coming back a year to these preserves that have moderate to high severe burns, you can see some of those, low to moderate burns. You can see a lot of that understory and grass being recolonized in those areas. So collaboratively and collectively, we have a lot of tools to bring to this scenario, but understanding that it really does take multiple ways of knowing and asking questions that speak to recovery, that speak of restorative narratives, I think is the power in this project, and is the power in my own pathway as an investigator. Concluding thoughts. I hope there are several Native students online, even if you're not. I would like to say that to all of you, there is an abundance of knowledge, of creativity, of innovation, for all of our cultures collectively, resources, critical thinking among us. And that I challenge you to tell those stories. I challenge you to think about what your headwaters are, all of those fundamental paradigms and values that you have, what tools are going to be useful in your toolkit, and which of those are most needed. And here's my acknowledgement slide. I think I'm done right on time. I really appreciate everybody tuning in. I appreciate those that are in the room with me today. All of the folks that have been planning stage for my visit here and making me feel really welcome. So I'll take questions if y'all have any.
Yeah, I have a question. Seems like you think that fungus have a beneficial effect. Is that correct?
Yeah, so what happens is that fungi form relationships with specific plant biota because they get from the plant a really stable carbon source, and in exchange help the plant access things like phosphorus and nitrogen.
Are those naturally occurring fungus, or do you introduce fungus? I mean, there's a lot of fungus.
Yeah, I breezed over a lot of those slides, but what we ended up doing in those experiments is isolating fungi that were collected from the sites. So we know which ones are there versus what is said to be there in the literature, right? So we tested 8 because some can be parasitic and some can be symbiotic. And we chose the ones that were symbiotic with these plants.
So the question I have, I'm kind of interested in fungus myself, but the question I have is, what's the mechanism, or why are the fungus beneficial? Do you have any idea like what they're doing?
Yeah, so fungi have a couple of things in addition to the plants. So fungi have what are known as phosphate transporters, but so do plants, right? And in addition to all of the small root hairs where you have like the most surface area and accessible to nutrients like phosphorus. So let me step back a second. Phosphorus and arsenic in this context are chemical analogs of each other. They have similar pKa's, they're similarly charged anions, oxyanions. So you might trick, right? If you have an abundance of both or you're replete in nutrients or deplete in nutrients, you can affect the way arsenic is uptaken into either the plants or the fungi. And how we're testing for the bioabsorption experiments is that all of the chitin, which are neutrally charged, could potentially bioabsorb the arsenate ions, or through those phosphate transporters inside of the plant, that could be a pathway.
Well, I don't want to manipulate everything, but one thing I know about fungus is that they excrete lactate. So that's what I was waiting to hear, something, that's
Yeah.
oxygen, powerful.
There are
oxygen
so many,
reduction pathways.
you're right. Yeah, you're right. There are so many like enzymatic pathways and even like reactive oxygen species that can be measured by spectrophotometry methods that we're proposing to do with UVV. So it's a really great point. Let me see. Fantastic presentation. Oh, thank you so much.
And then I'm just gonna announce, so we have a student-led Q&A at 4 o'clock. So we have about like maybe time for like 1 or 2 more questions here. And then students are welcome to stay and ask some more questions.
I'm reading the comments.
So if the plants are absorbing the arsenic, that means they can't. And is the idea here to, after a burn or a impact to the area, to plant a certain type of grass?
That's a really good question. That becomes like more an application, right? So you could essentially, what we figured out from these experiments and those without fungi are accumulating in the roots. And if you're growing these plants to feed cattle, or if you're growing these plants for like sweetgrass, where you harvest the above ground part and you burn and you're inhaling that, it says something about management strategies for planting a particular part or harvesting a particular part of a plant where you see the accumulation in the root system versus translocating into the above parts, which are used by people and by animals. So yeah.
There's time for one more question.
Or question.
Sure.
So on your work on the aerobic and aerobic arsenic, what's the role of the glucose in the aerobic?
It's an electron donor. We found it to be a really simple, cost-effective, thermodynamically favorable electron donor.
So you show the siding between full anaerobic and then aerobic anaerobic, right?
Right.
So what happens in the aerobic?
So what we're calling that water labile release. So it's very minimal. I think the most that we saw was like 5 micromolar compared to even those that were amended with glucose, similar within each other's standard deviation. So even if you give the microbes glucose and you give them oxygen to respire, they're not going to be forced to utilize those other metals in the redox tower that are similar, like iron, manganese, arsenic. So yeah. I think that's
So
it.
close us out. So I just wanted to say thank you to Dr. De Vore. It's been awesome. I've gotten to take her on campus today, so it's been really great to talk with you and take you around. I want to thank everyone who joined today and all the co-sponsors. And I would also like to thank Indigifuse, Sloan Indigenous Graduate Partnership, and the Chemical and Engineering Department for this hour of learning and growing together. And again, like I said, students are welcome to stay for our student-led Q&A beginning at 4. And please join us at the next seminar on March 14th at 3 p.m. to hear Dr. Otakoye Conroy Batten, who is a faculty in School of Sustainable Engineering and the Built Environment at Arizona State University. I believe that there's going to be a link in chat, yes, for the Native Voices in STEM website. And yeah, thank you again for everyone for coming. And if you want to clap it out again for Dr. De Vore, I'd appreciate that. Sorry, this is like awkward.